Hello everyone, let's talk about introduction and basic fish genetics. We have 6 questions in this topic. First one is, explain the importance of breeding program and its importance in aquaculture. The importance of breeding program is to increase production and product quality, increase product and cost efficiency, maintain genetic diversity, support the conservation and use of specific breeds, and consider animals' welfare and sustainable system. Because breeding program is to develop animals which more efficiently utilize available feed, land, and water resources. Second is, explain the principle and Hardy-Weinberg law. The principle of Hardy-Weinberg law is genetic variation in a population will remain constant from one generation to the next in the absence of disturbing factors. The condition which required for Hardy-Weinberg law is a large breeding population, random mating, no change in allelic frequency due to mutation. No immigration and immigration, it means, does not come into population because immigration and immigration can't alter allelic frequency. No natural selection of we select some individuals on the others will are selected are more common selective with the others. Third question is, explain the function of all the parts of cell. Part and function of cells. First one is cell membrane. Is the outer edge of the cell and forms the boundary between the inside of the cell with all of its organelles and the extracellular matrix. Next is nucleus. Has two main functions. It contains all of the oxyribonucleic acid of the cells and it's direct in the activities of the cells. Next is nucleolus. It's a small area with the nucleus where ribosomes are made. Ribosomes are described further down in this article. And nuclear membrane is similar to the cell membrane except that it surrounds the nucleus within the cell and perform less of a regulatory function. Cytoplasm or cytosol. The cytosol is a thick gel-like fluid that fills the space inside the cells in which the organelles are suspended. Endoplasmic reticulum is composed of interconnected membranous channel called cirrinae and is connected to the nuclear membrane. Next is Golgi apparatus, also made of systemites that are not interconnected. It takes molecules produced by the cells such as protein and lipids, modify them if necessary such as folding for proteins, and fax them into vesicles so that they can be shipped around or outside of the cell. Next, ribosome are organelles made of ribonucleic acid and protein and either attached to the endoplasmic reticulum or suspended in the cytosol. Next, in mitochondria are large organelles that have both an inner and outer membrane as well as their own mitochondrial DNA. Centrioles or centrosome. Centrosomes contain centrioles. The centrioles are a small gathering of microtubules that help with cell division during mitosis. Cytoskeleton is composed of a network of filaments and tubules that allows the organelles of the cells to remain in place and give the cell strength and shape. Baculus are small storage pockets formed in a single membrane layer containing gas such as oxygen or carbon dioxide, or fluids such as water found within cells. Vesicles are similar to baculus but are the part of transportation system of the cells. Lysosome. Lysosome are specialized vesicles in which protein enzymes are contained. The lysosomes break down macromolecules into their components for further use by the cell. Peroxisome. Peroxisome are the common in animal cells in perform oxidative digestion. Number 4 is the explain the central dogma of molecular biology, protein synthesis. The central dogma of molecular biology states that DNA contains construction for the making of protein, which are copied by RNA. RNA then uses the instructions to make a protein. DNA or oxyribonucleic acid contains the genes that determine who you are. Protein in turn determine the structure and function of all your cells. It begins with the sequence of amino acids that make up the protein. Ribonucleic acid, RNA, is a small molecule that can squeeze through pores in the nuclear membrane. It carries the information from DNA in the nucleus to a ribosome in the cytoplasm and then help assemble two protein. In short, DNA to RNA and RNA to protein. Question number 5 is explain principle of mitosis and meiosis. The principle of mitosis is somatic growth in plants and animals take place by the increase in the number of cells, and the meiosis, the pair of homologous chromosomes are divided into half to form haploid cells. In this separate or assortment of homologous chromosome is random. Meiosis and mitosis are both preceded by one round of DNA replication. However, meiosis includes the nuclear division, and 4 daughter cells resulting from meiosis are haploid and genetically distinct. The daughter cells resulting from mitosis are diploid and identical to the parent cell. Question number 6. Cite an example of a Mendelian inheritance of fish and gene action in fishes. Example, there are 2 different color switches gene in each fish. In this example, each there are 2 more sub-switches and can be either both on glow, meaning the presence of red color, but of glow, meaning no color, no one, or one of glow red, glow, which produce the same color as both were on. Example, the red fish and yellow fish combination are getting an orange fish. And that's all. Thank you.